I need to make a contraction of my presentation. But I want, anyway, to remember that we are using the more instrument for the cell impact. And we have to remember that we have natural and artificial electromagnetic fields. Natural are not pulsed, but artificial are pulsed. That's the biological impact, the difference between the two. The human evolution were in a RF-free environment. The difference, for example, from the low limit are this 0.0002 volt per meters in the environment. ICNIRP limit 61 volt per meter, Italy 6 volt per meter. You can see the enormous difference between the environment in which we evolved and the environment in which we live in the town, for example. Natural electric field is high, 130 volt per meter, and we have protected during the evolution by this electric field all the our next nervous system is protected by myelinic cells, which made a screen, a shield for the natural electric field to protect the nervous signals that go on through our body. Next. Magnetic fields also is has a very great importance. Is 50 microtesla. And you can remember that the pulsed magnetic fields at 50 Hertz can double the risk for leukemia. The pulsation is the difference between the two. Go on. I say yes, the electromagnetic fields in the environment is like put a bottle of ink in an aquarium, and we are the fish inside. The artificial electromagnetic fields comes from the power lines, for example, or by use of electrodomestic. If you make these experiments, you take an oscilloscope and touch the probe of the oscilloscope, you can see that your body is pulsing at 50 Hertz like the current which we can use in the houses. And this pulsation is a stress for the body. Few people remember that power frequency was declared human possible carcinogen from YARC in 2001. Nobody says that it's dangerous to stay near the electric oven in home. Next. This electromagnetic spectrum in which you can notice that the electromagnetic waves changes of wavelength in changing their frequency. And for the human effects and the body effects, we can notice that different frequencies has different influence. low frequencies, for example, 50 Hertz, or low frequency again, like 10 to 40 Hertz, can move ions inside the cells. We use, for example, this kind of approach to differentiate stem cells in our institute. Next. High frequencies have this aspect in many places in the country, again. And if we can measure analysis of the electromagnetic spectrum in different houses, you can see the first one in which in a home of an electrosensible person, there is a pointer, please. And in the low spectrum, you can see a house which is not irradiated. So it's simple to understand that there is a very big difference in irradiation from these two houses, and this is why the person which lives in Cinecittà, which is the first spectrum, is ill from different symptoms. This reported reversible symptoms near the telephone antennas, as reported by Wolf and Wolf in 2004. Next. And we have to remember also that IARC in 2011 classified the radio frequency as a possible carcinogen. The problem of thermal and non-thermal effects is quite not a problem, because in this monograph published in 2010 by Ramazzini, Dr. Giuliani and Soffritti, is summarized a number of 158 studies which report non-thermal effects and mechanism of the radio frequency. I remember here that in 2002 we found an ICEMS International Commission for the Electromagnetic Safety, and you can recognize the most important independent scientists in the world that participated in that commission. No. Sometimes the telephone is used as babysitter. And in 30 years of research in our institute, I want to remember some studies that we made, and some in publication now, and some published in different years. This report, for example, the genetic effects of radio frequency at 900 megahertz of telephone in cultured cells. As you can see, the green line reports the activation of pro-apoptotic, so the cell death signals, in the cell after 2 hours of exposure. And this signal, this regulation signal of DNA, became less during time, while a pro-survival signal for example, RAS and the other controller of DNA, became higher in the time. So this is a dangerous thing because not only the telephone signal can kill the cells, 20% of cells were killed in these experiments, but the survival signals can make proliferate, induce proliferation of cells, also of of the cells which were partially damaged during the first phase of the experiments. We thought that was the time to make these experiments not only in the lab, but also in the house in which the person were irradiated from the environmental signals. Prepared an incubator for cells like this, in which you can see the flask of cells irradiated and flask of cells protected from radiation as control cells. And we made different experiments near radar emission station, for example. Next. You can see here the radar signals, the cells protected, the antenna for see the radar inside the incubator and so on. Go on. And we made, for example, experiments of cultivation of cell in different houses in which there were patients and persons which had different illness, probably due to the radiation. Next. And one simple test to see if there are damages in the irradiated cells is the viability test, MTT is the name, in which you can join a staining substance, which is a metabolic substance for the cells, which became colored if they are live and don't metabolize the staining if they are dead. As you see in these preparations, you can see the control cell which are in dark well, and the exposed cells which are not metabolized the staining, so they are dead. This experiment is an example of the experiment we can do. We made this also at 5.8 gigahertz in a farm. Next. The animals in the farm has different pathologies from quantity of milk and illness. Next. This is the system to cultivate the cells inside the farm, near the animals. Next. And here also you can see an example of the difference between the exposed cells and the control cell not exposed. Next. Radio frequency damages at home. Near the antenna, of the telephone antenna in a little country in Italy, next, we used the same type of cultivation system, next. And we can see the different wavelength of the signal present in the house, next, and the spectrum analysis of that signal. And here also we can see the difference. The fields inside the house, which were used for the experiment, were 1.6, 0.9, 2.1, 1.6 volt per meter. And this is the difference between is an example, naturally. We made also statistical measurements about that, in which the cells exposed like this are not metabolized the staining, while the control cells are colored. This difference is due to the gene effects. Next one. This is the gene expression of different genes, the N-methyltransferase, UHRF, and caspase, which is the death gene, which are more exposed, for example, in the exposed cells, and less present as a controller gene in the control cells. Again. In a graphical condition, you can see better the different expression of the genes which control in different kind of cells the control of the genes, and in which is possible to see the difference induced by the electromagnetic fields in the houses. Next. We use also different approach to make research with the Wi-Fi pulsation. Wi-Fi is a pulsation at 2,437 megahertz. But the difference between the signal of a telephone, for example, is that, next, the bandwidth of the signal is full by peaks which are emitted for data transport. This made the Wi-Fi signal very similar to the radar signal, because the impulse take and bring the data through the bandwidth. And every peak of emission is an inducer inside the cells of electricity, which is not previewed from the cells. Next, again. We found also different in some changes in enzymatic kinetics in glutathione and catalase, which are two enzymes important for the person which are electro hypersensitivity or chemical sensitivity. Again. Next. In this curve, you can see the difference of the irradiation of the enzyme by Wi-Fi wavelength, in which you can see the difference of energy used by the native enzyme, not irradiated, and in blue, the irradiated enzyme, which need more energy to function. So the difference is the less function of the enzyme inside the cells of the exposed person. Again, we made also some experiment with dosimetry in electrosensitivity persons. And we found an interesting thing. This person always are treated as fools, which are not very in health. Sometimes some medical doctor also speak about idiopathic illness, which has no significance. And they are not neurological persons. So we try to give to they an exposure system, a recorder of the exposure, which record the exposure, and they have the possibility to insert a marker when they have symptoms, and to notice a diary of the symptoms, in which was possible to connect, to make a correlation between the symptoms and the irradiation. You see, for example, here, the marker put from the person when he have the symptoms. Next. This is the diary annotated again. And you can see that the symptoms were inside the radio frequency radiation recording. So this is simple to make a correlation between the symptoms and the irradiation. But there is also another interesting thing. Analyzing statistically the data from the recording, we found that if the record report the radio frequency of 900 megahertz, the patients have a delay from demonstration of symptoms of 15 minutes. If they were irradiated from 1,800 and 1,900 megahertz, they had a delay from to the symptoms of 3 minutes, and 2,200, 15 minutes of delay, Wi-Fi 2,400, 10 minutes delay. So is sometimes can explicate if what happens when, for example, you make a telephone call and have some headache about 1 quarter later. This is differences in ancient antennas and the next emitters for 5G. These are ray emitters which use, for example, in this type, 84864 emitters, which collaborate one with the other to construct the fields of 5G. And they function from 24 to 39 gigahertz. Next. The Federal Commission Communication in the United States, its own will, says 5G is so big business that we no use standards, no testing for people. Next. I want to remember that there was a publication of Yuri Feldman in 2008 in American Physical Society, which demonstrated that the epidermis has sweet duct which are in form of helicoid. Next one. And this kind of antenna is a perfect reception, receptivity antenna for the millimeter waves. And we have 100 sebaceous glands for square centimeter of the skin. And the skin is accepted by the 5G radiation. For example, in the gland duct, as I say before, and in the nerve termination, which are in the first part of the epidermis. You know, you touch, and the nerve conduct the signal to the cerebral spinal and the hippocampus. And what happens is that hippocampus connected to the memory and to the emotional. And after this elaboration, the signal go to the cortex, which elaborate the response. So 5G is possible, an exception of the nervous system through all the epidermis. So the damages are well known from radio frequency and electromagnetic field. We have to use a prevention principle, not only precautionary principle. Next. What we can do is very difficult. No Wi-Fi at home, at school, only by cable, all via cavo. Only by cable the signal should be used. Next. This is, in Italy, I asked for a campaign for the cellular phone strike the first day of the month, which is active since June. The problem is the telephone first. If you don't use, no antenna needed. Cell telephone is an emergency radio that, to use only in emergency, not to substitute the normal communication, which should be done by cable. Next. Thanks for your attention.